Domagoj Gajski with assessment of the biocontrol potential of natural enemies of the psyllid populations in a pear tree orchard during spring.
Thank you for the introduction. Give me a second. Okay, perfect. Now let me just minimize this so that I can see. Okay, perfect. Good afternoon, everyone. My name is Domagoj Gajski. I come originally from Croatia, but currently I'm performing my PhD studies at the Maastricht University in Brno, Czech Republic, where I study trophic interactions of arthropod communities inside agroecosystems. And today I will talk to you about my research, which was recently published in Pest Management Science, where we assess the biocontrol potential of natural enemies against psyllid populations in a pear tree orchard during spring. To start with, I will explain what are psyllids. These are hemipterans, which are the main pest in pear orchards in most of the world. They make economical damage by extracting nutrients from pear trees by sucking the phloem of the leaves. And they indirectly induce development of sooty molds on pear trees. And they as well transmit harmful phytoplasms that cause pear decline, sometimes even pear deaths. And the main problem with them actually started some 50, 60 years ago when we introduced non-selective pesticides in pear production. And those non-selective pesticides actually induced resistance to those pesticides in the psyllids because of their polyvolatile nature. They, in the meantime, as well, let's say, killed or reduced the number of all other naturally occurring arthropods in the orchards. And another issue with those facilities that they are active already in early spring. They already start hatching eggs when the first leaf buds start to burst. And this means that their numbers can be quite high in the beginning of the summer. So nowadays, scientists are more interested in other approaches than the chemical control of these pests. They try to use a conservative biological control. One of the methods is using naturally occurring predators. The most famous predators which is used is the specialist Anthochoris bugs. They are famous because they are, let's say, specialized on feeding on psyllids. And the only issue with them is that their abundances are usually low over the whole season. So they tried some approaches like augmentative approaches where they artificially increase their numbers and then release them in the orchards. And this way they tried to, in some regions, control. Somewhere it was effective, somewhere less. And more and more nowadays, we want to focus on the generous predators, which are much more abundant in all these orchards, especially in Central Europe, for which we really still don't have any info about how frequently, although they are generous, how frequently they feed on the main pest. Therefore, we decided to investigate the community of naturally occurring predators in an abundant pear orchard during spring. We wanted to evaluate their contribution towards the control of psyllid populations. And we did that by using the agnostic PCR to speed up the research, because by observing it by naked eye, it will be quite time consuming. So we decided to choose an abandoned pear orchard near Brno, Czech Republic. Why abandoned? Because we didn't want to have any side effects of any kind of pesticides which were used there. We sampled once a week from early spring to early summer. Each week we sampled from 25 different trees. One sample was 5 trees, so we chose 5 different rows of the orchard. We sampled by beating branches and collecting everything that falls into the net. And then we stored all the predators and the psyllids into ethanol. And then later on in the lab, we identified them and calculated the abundance. Later, we performed diagnostic PCR, where we first extracted the DNA from the abdomen of the predators, as you can see here on the right picture, where we extracted the abdomen of a clubionid spider. After that, we performed PCR by using Calcopsilla-specific primers, which we designed. And here on the left, you can see a picture of our results on the gel electrophoresis, where the result was when we saw a band. This means that a psyllid was recently eaten by this sample. And if there was a negative result, this means that there was no recent feeding of a psyllid by this predator. And we, of course, as well use positive negative controls so that our protocol that we were confident the protocol was working. And here are the results. We found 4 main distinct groups of predators in the orchard. The most abundant, the most diverse were definitely the spiders, and I'll talk about them a little bit later. The second ones were the lady beetles. They had around 7 species in our orchards, and the most abundant were by 80% the Asian ladybug or Harmonia Psyllidis. Then we had as well this already mentioned anthochorid bugs, although their numbers were really low in the spring. We found over the whole spring season only 48 individuals. And we as well had cantharid beetles, which had only 37 individuals. So we decided to exclude them completely from further studies because they were not so interesting to us. And then we calculated the relative frequency of positive individuals. We found out that, as previous studies have shown, anthochorid bugs are quite specialized for psyllids, as we found prey DNA in 90% of all the individuals we collected. Although due to their low number, we can't really say what is the high potential there. Then what was really surprising to us was that coccinellid beetles had a surprising low frequency of positive individuals, although of their high abundance. We then decided to look at spiders, which had a little bit larger frequency. And we found out that they have 4.7 times more positive individuals than, for example, anthochorid bugs. So we decided to focus more on the spiders. And there, inside the spider group, we found several families. And we as well found out that actively hunting spiders had a higher predation frequency on psyllids compared to, for example, web-building spiders, which are these dictinids, pteridids, or ambushing spiders, stomicids. And out of all the actively hunting spiders, we found that philodromids had, of all the species we could find, the highest amount of positive individuals. So there should be more focus on those spiders, definitely. Lastly, we wanted to compare and see if there is a significant correlation between the abundance of psyllids and abundance of any kind of predators to see if there is a numerical response to the abundance. We found out that only the abundance of psyllids had only a significant effect on the abundance of spiders. So that when the abundance of psyllids increased, so did the abundance of spiders, but it increased asymptotically. So there was a limit to it, mostly due to the territorial behavior of spiders. However, as we can see here on the chart, on the left side of the chart, most of the data of the abundance was on the left side of the chart of the psyllids. So this means actually that this part of the chart, they can really control perfectly. They can, let's say, numerically respond to the abundance really easily. With everything said, I want to conclude that anthochorid bugs had the highest frequency of positive individuals. However, their overall abundance was low in spring. So we could only use them if we artificially augment their numbers in the orchard. Coccinellid beetles were, to our surprise, really low in abundance. So actually very high in abundance, but low in frequency. So it seems that they are more keen to aphids, which are as well common in those orchards. But what was the most interesting fact is that foliage-dwelling spiders were by far the most abundant predators, which was shown as well in other studies. But they had as well the high overall predation rate, and they showed a numerical response to the psyllid density. And with everything said, we feel that there is a high potential in using those spiders as biocontrol agents in the spring in orchards in Central Europe. And future studies should tell us more about if we can use them in controlling these annoying pests. Thank you very much for your attention. And if you have any questions, feel free to ask.
All right. Thank you very much. We have time for questions. All right. So Sven Weber asks, even though the main focus lies on spiders, did you look at coccinellids, larvae, and adults? And if so, do they behave differently as biocontrol?
Yes, definitely. I didn't mention here because I think it will be just taking too much time, but we found as well and studied as well the larval and adult stages of coccinellid beetles. And we really didn't find any significant difference between these stages in the diet and in the preferences for the psyllids. Although they definitely have, how to say, they definitely have I mean, how to say, for example, the adults ones have a behavior of flying, while the larvae are just crawling around in the trees. We really didn't find any significant difference.
Susan Kennedy compliments your talk and says, what is the scale of the taxon specificity of your psyllid primers? That is, did they amplify all psyllids or just some?
Mm-hmm. So I tested my primers on the leg sample of every single arthropod specimen we could find in the orchards. There were around 80 different species. And they didn't amplify any other samples except the three psyllid species which we found in the orchards. Those were Cocopsilla piri, Piricula, and Pirisuda. So they are, let's say, genera specific. Yeah.
And Sean McCann asks, how long after a meal is the DNA amplifiable?
That's a good question. That's a good question. There have been a lot of studies, and there is a, let's say, slight difference between different predators in the detection of the prey DNA in it. For example, I think the lowest, actually, let's say the fastest metabolism we found in previous studies has been found in anthochorid bugs, for example, and spiders had the let's say lowest metabolism, so the DNA could be found much longer there. However, as I said, we decided to therefore we decided actually to sample, let's say, on a weekly basis, so that we have let's say reduce this bias of these connections. And as well, even we made a calculation based on a previous paper on reducing these biases depending on the time it's needed to devour the DNA. And it found that still the spiders were the most significant predators. They had the highest number of positive individuals if we reduced it with the time period. Anyway, if we corrected it on the time period of degradation.
Great. Thank you. Are there any more questions for Domagoj?
I think I see one more, yeah.
This is one last one from Sven. Was there a difference between adult and juvenile spiders in the group shown?
Yeah. So most of the spiders which we found there in spring were subadults. There were sometimes some adults already, let's say for their philodromids later were already some adults, but there were not so many. So most of the species were subadults. Or either subadults or juveniles. Yeah. So we just focused on them, and I didn't have, let's say, a larger sample size of any of these species, let's say, of adults that I could see, let's say, a difference between. Yeah.
All right. Great. Thank you so much.
Thank you very much.
So the